Hello there, and welcome to the Deep Sleep Science channel. I'm so glad you've joined me tonight. Beneath every forest, under every footstep, a hidden kingdom spreads through the dark earth in quiet threads. Most of it will never be seen. Most of it has no name. Fungi live between worlds, neither plant nor animal, neither fully visible nor completely hidden. They are the soft architects of decay and renewal, the gentle recyclers of all that falls. For billions of years, they have been breaking down the old to make way for the new. They breathe like we do. They communicate through vast underground networks that stretch further than we can imagine. Some glow softly in the dark. Others can survive the vacuum of space. Science is only beginning to understand the scale and strangeness of the fungal world. And what we're discovering is as calming as it is extraordinary. Tonight, we'll walk gently through 100 quiet facts about fungi, facts that remind us of patience, connection, and the slow, invisible work that sustains all life. If you enjoy these journeys, consider liking and subscribing, or share your thoughts in the comments. It helps others find their way here too. But for now, there's nothing you need to do. Just let yourself settle. Let your breath slow, and let the hidden world of fungi open like a dark forest floor after rain. Now, if you're ready, let's begin. Fungi form their own kingdom of life, separate from plants and animals. Until the middle of the 20th century, we grouped them with plants in textbooks and field guides, placing mushrooms alongside ferns and mosses in a category we called the cryptogams, the hidden reproducers. But as our microscopes grew more powerful and our understanding of cellular chemistry deepened, it became clear that fungi were something else entirely. They do not capture sunlight and convert it into sugar the way plants do. They do not build their tissues from cellulose. Instead, they secrete digestive enzymes into their surroundings and absorb the nutrients that result, dissolving the world around them before drawing it inward. In the middle of the 20th century, scientists formally recognized what had been slowly becoming apparent. Fungi constitute their own kingdom, governed by different principles, operating according to different metabolic rules. It was an acknowledgement that life's diversity cannot be contained within simple categories, that between the green world of photosynthesis and the mobile world of animal hunger lies a third realm, ancient, patient, and fundamentally distinct. They are more closely related to animals than to plants. This fact tends to surprise people when they first encounter it, because fungi seem so plant-like in their stillness, in the way they seem rooted to one place. But molecular phylogenetics, the study of evolutionary relationships through DNA and RNA sequences, has revealed a hidden kinship that contradicts appearances. Animals and fungi both belong to a larger group called the Opisthokonts, a name derived from the Greek words for rear and pole, referring to the single posterior flagellum that their common ancestor possessed. Plants, by contrast, diverged from this lineage long before, following an entirely different evolutionary path. The evidence for this relationship is written in countless molecular details. Fungi and animals share similar ribosomal RNA sequences, similar protein structures, similar mechanisms for regulating cell division. Both groups are heterotrophs, meaning they must consume organic matter rather than synthesizing it from sunlight. Where plants build, fungi and animals both consume. It is a kinship that operates beneath the surface of form, a relatedness etched not in shape, but in the fundamental chemistry of cells. Fungi and animals shared a common ancestor over 1 billion years ago. To grasp the vastness of this timescale, consider that 1 billion years ago, the Earth had no forests, no flowers, no insects or birds or mammals. The continents were arranged differently. The atmosphere contained less oxygen. The oceans teemed with single-celled life that had not yet learned the complex choreography of multicellularity. Somewhere in those ancient seas, a microscopic organism existed that would eventually give rise to two wildly different lineages. One that would develop the capacity for movement, for sensation, for thought, and another that would master the art of quiet decomposition. The divergence between animals and fungi represents one of the great branchings in the tree of life, a split that occurred so long ago that mountain ranges have risen and eroded away in the time since, that entire oceans have opened and closed. Recent genomic studies have shown that the paths leading to animals and fungi were not only divergent, but followed contrasting trajectories even before the origin of either group. Animals accumulating genes for multicellular coordination and communication. Fungi streamlining their genomes and focusing on metabolic efficiency. And yet, despite 1 billion years of separate evolution, we still carry traces of that shared origin written in our DNA, faint molecular echoes of an ancestor we can never see. Like us, fungi breathe in oxygen and exhale carbon dioxide. They are aerobic organisms, relying on the same respiratory process that powers your heartbeat and fuels your thoughts. Unlike plants, which take in carbon dioxide and release oxygen during photosynthesis, fungi consume oxygen and release carbon dioxide as a byproduct of cellular respiration, just as we do. This means that beneath the forest floor, within every rotting log and every square inch of soil, there is a vast unseen respiration occurring. The mycelium threading through the earth is inhaling and exhaling in a rhythm so slow and subtle that it operates entirely outside our awareness. Walk through a woodland and you are surrounded by breath. Not only the breath of trees cycling gases through their leaves, not only the breath of insects and birds and deer, but the breath of countless fungi woven through the substrate beneath your feet. It is one of life's most profound symmetries. We breathe the same air. We rely on the same chemical exchange. We are bound together in a single atmospheric cycle that connects the mushroom to the human in ways we rarely pause to consider. Fungal cell walls contain chitin, the same material found in lobster and crab shells. While plants construct their cellular boundaries from cellulose, a polymer made of glucose molecules linked in long chains, fungi build theirs from chitin, a substance composed of N-acetyl glucosamine units arranged in crystalline microfibrils. Chitin is one of nature's most versatile structural materials, providing both strength and flexibility. It is what gives the exoskeletons of insects, arachnids, and crustaceans their remarkable durability, allowing a beetle's shell to withstand forces many times its body weight, allowing a lobster's carapace to protect it from the pressures of the deep ocean. The presence of chitin in fungal cells is yet another molecular signature of their evolutionary kinship with animals, a shared chemistry that links the mushroom sprouting from a forest floor to the crab scuttling across a tidal pool. When you run your fingers over the smooth surface of a mushroom cap, or the tough, leathery texture of a bracket fungus growing on a tree trunk, you are touching chitin, the same substance that armors the creatures of the sea and the insects that hum through summer air. Scientists estimate there are between 2.2 and 3.8 million species of fungi on Earth. This remarkable figure is not a count. but an extrapolation, derived from decades of careful comparison between fungal and plant diversity ratios, from environmental DNA sequencing studies that detect fungal genetic material in soil and water samples, and from statistical models that attempt to account for the vast number of species that have never been directly observed. The range itself, spanning more than 1.5 million species, speaks to the profound uncertainty inherent in cataloguing a kingdom where most members live their entire existence as microscopic filaments hidden within substrates we rarely examine. Each habitat that has been intensively studied reveals layer upon layer of fungal diversity: soil fungi, wood decay fungi, fungi that live as parasites within plants and insects, fungi that form partnerships with algae to become lichens, fungi that exist only as single cells floating in aquatic environments. And environmental sequencing studies, which extract and analyze DNA directly from samples without needing to culture or even see the organisms, consistently reveal that the majority of fungal diversity remains completely unknown to science, existing in a taxonomic shadow where DNA sequences have been detected, but no name has been assigned, no description written, no life history documented. Only about 148,000 fungal species have been formally described and named. Even using the more conservative estimate of 2.2 million total species, this means we have identified perhaps only 6 or 7% of the fungal diversity that exists. The vast majority of the fungal kingdom remains scientifically invisible, carrying on the work of decomposition, symbiosis, and nutrient cycling, without names, without entries in taxonomic databases, without any formal acknowledgement from human science. There are fungi growing in your local woodland that belong to species never documented in scientific literature. There are yeasts living in tree bark, molds decomposing leaf litter, and microscopic fungi colonizing plant roots in your own backyard that have no place in any field guide ever published. They exist in a realm beyond language, beyond classification, performing their ecological roles with the same patient efficiency as the species we do recognize, untroubled by our ignorance of them. The largest living organism on the planet is a fungus in Oregon, called the humongous fungus. It is easy to assume that the largest living thing on Earth would be something visible and monumental, a giant sequoia reaching toward the sky perhaps, or a blue whale gliding through the ocean depths. But the true record holder lives mostly out of sight, woven through the soil of the Malheur National Forest in eastern Oregon. It is a specimen of Armillaria ostoi, a honey mushroom that spreads not upward but outward, threading its way through the forest floor in a vast underground network. Scientists discovered its extraordinary extent in the late 1990s by collecting tissue samples from mushrooms scattered across the forest and testing whether they would fuse together in laboratory dishes, a sign that they belong to the same genetic individual. DNA fingerprinting confirmed what the fusion test suggested. What appeared to be many separate fungi were in fact a single organism, one continuous living entity stretching across the landscape in silent, patient expansion. This single fungal organism spreads across more than 2,400 acres of forest floor. To grasp this scale, imagine nearly 4 square miles of woodland, an area large enough to contain more than 1,800 football fields laid side by side. The fungus occupies this vast territory, not as a single visible mass, but as an intricate lattice of filaments spreading through soil and beneath bark, connecting to tree roots, breaking down wood, slowly digesting the forest from within. It advances at a pace measured in inches per year, somewhere between 8 inches and 3 feet annually, a creeping expansion so gradual that individual trees may live for decades before succumbing to its slow parasitism. The organism weighs an estimated 35,000 tons, making it heavier than the largest blue whales, heavier than entire herds of elephants. And yet, despite this immense mass and extent, most of it remains invisible, buried in darkness, perceptible only in autumn, when honey-colored mushrooms bloom from the forest floor like scattered signals from a hidden continent. The humongous fungus is estimated to be thousands of years old. Based on its current growth rate and geographic extent, scientists believe the organism is at least 2,400 years old, though it may be as ancient as 8,650 years. If the older estimate is correct, this fungus began its life during the Bronze Age, spreading its first filaments through the soil when humans were just learning to work iron, when mammoths still walked the earth in isolated pockets. It has persisted through millennia of seasonal cycles, thousands of winters, thousands of springs, growing slowly outward in all directions. A living memory of deep time written in mycelium rather than in stone. Trees have sprouted, matured, and fallen within its territory. Fires have swept through the forest. Ice ages have come and gone. And through it all, the fungus has continued its quiet work, absorbing nutrients, spreading through roots, releasing spores into autumn air. It is a reminder that life's grandest expressions are not always the fastest or the most conspicuous, but sometimes the slowest and most patient, the ones that measure time in centuries rather than seasons. Mushrooms are not the fungus itself. They are the fruiting bodies like apples on a tree. This is one of the most important and often misunderstood facts about fungal biology. When you see a mushroom pushing up through leaf litter or growing on the side of a log, you are not seeing the organism in its entirety. You are seeing only its reproductive structure, a temporary organ produced for the sole purpose of creating and dispersing spores. The analogy to an apple tree is imperfect but useful. Just as the apple is not the tree, but merely the fruit it produces, the mushroom is not the fungus, but the fruiting body that emerges when conditions are favorable, and the organism is ready to reproduce. The tree itself, trunk, branches, roots, persists year after year, while the apples appear seasonally and then decay. In the same way, the true body of the fungus lives on beneath the surface, and the mushrooms we see are ephemeral structures that may last only days or weeks before withering away. The fungus endures. The mushroom is merely its fleeting expression. A brief gesture toward the future in the form of billions of microscopic spores. The main body of a fungus is a network of threads called mycelium. These threads, known individually as hyphae, are extraordinarily fine, often only a few micrometers in diameter, far thinner than a human hair. They branch and rebranch as they grow, forming a diffuse, cobweb-like structure that permeates whatever substrate the fungus is colonizing: soil, wood, leaf litter, even living tissue. Mycelium is the fungus's feeding apparatus, its sensory organ, and its means of exploration all in one. Through these delicate filaments, the fungus secretes digestive enzymes into its environment, breaking down complex organic molecules into simpler compounds that can be absorbed back through the hyphal walls. The mycelium is constantly growing at its tips, probing forward into new territory, seeking nutrients and moisture, responding to chemical gradients in ways that resemble though they are not, a kind of intelligence. It is a structure both fragile and resilient, easily broken but quick to regenerate, capable of spreading across vast areas or remaining confined to a few cubic inches of substrate. A single teaspoon of healthy soil can contain up to 8 miles of fungal filaments. This fact is almost impossible to visualize. 8 miles of living thread compressed into a volume no larger than the hollow of a spoon. It speaks to the extraordinary fineness of hyphal strands, which can be packed together in near microscopic density while still maintaining their function. These filaments do not lie passive in the soil. They are actively growing, secreting enzymes, absorbing nutrients, forming connections with plant roots, communicating chemically with bacteria and other microorganisms. The soil is not empty space scattered with particles of rock and organic matter. It is densely inhabited, a living matrix where roots and fungi and bacterial colonies intermingle in relationships of staggering complexity. And fungi are among the most pervasive inhabitants of this hidden world, their mycelial networks threading through every gram of healthy soil, binding it together, enriching it with organic compounds, facilitating the flow of nutrients between organisms. If you lined up all the mycelium in 1 teaspoon of soil, it could stretch up to 10 kilometers. 10 kilometers, more than 6 miles, of fungal tissue coiled within a space small enough to rest on your tongue. Globally, researchers estimate that the total length of fungal mycelium in just the top 10 centimeters of Earth's soil exceeds 450 quadrillion kilometers, a distance so vast it is roughly half the width of the Milky Way galaxy. These numbers are almost too large to hold in the mind. They collapse under their own immensity into something that feels more like poetry than science. But they point toward a profound truth, that the world beneath our feet is far more intricate, far more densely inhabited, far more alive than we tend to imagine. Every step you take is supported by an invisible architecture of fungal filaments, a living scaffolding that holds the soil together and mediates the passage of water and nutrients through the earth. Mycelium spreads underground in branching web-like patterns. The growth habit of mycelium is fundamentally different from the growth of roots, though the two are often compared. Where roots tend to grow in relatively straight lines, extending downward or outward in search of water and anchorage, mycelium branches prolifically, sending out side hyphae that themselves branch again and again, creating a fractal-like network that maximizes surface area. This branching architecture allows the fungus to explore a large volume of substrate efficiently, to respond rapidly to localized pockets of nutrients, and to reroute resources through the network as conditions change. Hyphae can fuse with one another, creating interconnections that allow nutrients and even electrical signals to flow through the mycelial network in complex patterns. The result is a structure that resembles a web in both form and function, a distributed, decentralized network with no single point of control, capable of growing, repairing itself, and adapting to disturbances with remarkable resilience. This network is sometimes called the Wood Wide Web. The term, a playful reference to the World Wide Web, was coined to describe the underground fungal networks that connect individual plants in an ecosystem, allowing them to exchange resources and information. These networks are formed primarily by mycorrhizal fungi, species that form symbiotic partnerships with plant roots, extending far beyond the root zone and linking multiple plants together through shared mycelial connections. Through these fungal intermediaries, trees can share water during drought, transfer carbon from well-lit areas to shaded seedlings, and even transmit chemical warning signals when under attack by pests. The metaphor of an internet is apt in some ways. Both are decentralized networks that facilitate the flow of information and resources across great distances. But the Wood Wide Web operates on biological timescales, through chemical exchanges rather than electrical signals, and its connections have been evolving for hundreds of millions of years. It is a testament to the interconnectedness of life, a reminder that what appears to be a collection of separate individuals, trees standing alone in a forest, may in fact be profoundly linked beneath the surface, sustained by relationships we are only beginning to understand. Fungi can connect the roots of different trees and plants through mycelial threads. These connections are not accidental, but intentional partnerships formed by mycorrhizal fungi, species that have evolved to form symbiotic relationships with plant roots. The fungus wraps its hyphae around or even penetrates into the fine root hairs of a plant, creating an interface where nutrients and water can be exchanged. The plant provides the fungus with sugars produced through photosynthesis, compounds rich in carbon that the fungus cannot manufacture on its own. In return, the fungal network extends far beyond the reach of the plant's roots, exploring a much larger volume of soil and accessing nutrients, particularly phosphorus and nitrogen, that the plant could never obtain alone. But the exchange does not stop there. Because a single mycelial network can connect to multiple plants simultaneously, these fungal threads become living conduits through which separate organisms can share resources. What appears above ground as a collection of individual plants is below ground a community linked by fungal intermediaries, bound together in relationships of mutual dependence. Through these connections, trees can share water, nutrients, and even chemical warnings. Research has shown that when one tree is well watered and another is experiencing drought, water can move through the fungal network from the hydrated tree to the parched one. Carbon compounds follow similar pathways, flowing from trees bathed in sunlight to seedlings growing in shade, from mature individuals with extensive photosynthetic capacity to younger plants struggling to establish themselves. Perhaps most remarkably, when a tree is attacked by insects or infected by pathogens, it can release chemical signals into the fungal network, defensive compounds that trigger neighboring trees to ramp up their own immune responses before the threat arrives. These chemical messages travel through the mycelium faster than they could diffuse through soil or air, providing an early warning system that allows plants to prepare their defenses in advance. The forest, it turns out, is not a collection of competitors struggling in isolation, but a network of allies sharing information and resources through fungal channels, coordinating responses to environmental challenges in ways that blur the boundaries between individual and collective. A single mycelial network can link dozens of trees across a forest. Studies using radioactive tracers and genetic analysis have revealed that individual fungal networks can span hundreds of square meters, connecting 30, 40, even 50 or more trees in an interwoven web of exchange. The largest trees in a stand, the oldest, the tallest, the most deeply rooted, tend to have the most extensive fungal connections, serving as hubs in the network through which resources flow to smaller, younger individuals. These hub trees, sometimes called mother trees, play a disproportionate role in the health of the forest, acting as anchors and reservoirs that stabilize the system during periods of stress. Remove one of these central individuals and the network fragments, leaving younger trees more vulnerable, less able to access water during drought or nutrients during lean seasons. The connections are not random. They show patterns and preferences, with trees of the same species often more densely linked to one another, though cross-species connections also occur. It is an architecture of interdependence, invisible from above, but essential to the forest's resilience. Mother trees use fungal networks to nurture their seedlings in the shade. This phenomenon, documented through careful field experiments, suggests a level of resource allocation that borders on the parental. Large, established trees pump carbon through mycorrhizal networks to their own offspring, seedlings that share their genetic material and are often growing in the deep shade cast by the parent's canopy. These young trees, unable to photosynthesize efficiently in low light, depend on the supplemental carbon they receive through fungal intermediaries to survive their vulnerable early years. The mother tree, in effect, subsidizes its descendants, allowing them to persist in conditions where they could not otherwise survive until a gap opens in the canopy and light becomes available. This transfer is selective. Isotope studies show that carbon flows preferentially to kin rather than to unrelated seedlings, implying that the forest possesses mechanisms for recognizing genetic relatedness, even in the absence of a nervous system. It is a quiet form of care, enacted not through movement or sound, but through the steady pulse of sugars, through fungal threads. A reminder that nurture exists in forms far stranger than we typically imagine. Fungi absorb nutrients from their surroundings rather than producing food through photosynthesis. This fundamental metabolic difference separates fungi from plants in the most essential way. Plants are autotrophs, self-feeders, capable of capturing sunlight and using its energy to convert carbon dioxide and water into glucose and other organic compounds. Fungi, by contrast, are heterotrophs, like animals, meaning they must obtain organic carbon from external sources. They cannot create food from light and air. They must consume what already exists, breaking it down into forms they can absorb. But unlike animals, which ingest food and digest it internally, fungi digest externally. They live inside their food, surrounded by the very substance they are consuming. It is a mode of nutrition unique to fungi and certain microorganisms, a way of being that dissolves the boundary between the organism and its environment, where feeding and dwelling become the same act. They secrete enzymes into their environment to break down complex organic matter. Fungal hyphae release a suite of powerful enzymes through their cell walls into the surrounding substrate, whether that substrate is wood, leaf litter, soil, or living tissue. These enzymes include cellulases that break down cellulose, ligninases that degrade lignin, proteases that cleave proteins into amino acids, and lipases that split fats into fatty acids and glycerol. Each enzyme is specialized for a particular type of chemical bond, and together they form a molecular toolkit capable of dismantling some of the most recalcitrant materials in nature. The broken down products, simple sugars, amino acids, minerals, are then absorbed through the hyphal walls and transported through the mycelium to wherever they are needed. This external digestion allows fungi to colonize substrates that would be inaccessible to organisms relying on internal digestion, to extract nutrients from materials so tough and chemically complex that nothing else can break them down. It is a patient, thorough process, operating on timescales of months and years, slowly transforming the solid into the soluble, the dead into the available. Fungi are nature's greatest decomposers, recycling dead plants and animals back into soil. In every ecosystem on Earth, fungi play this essential role, quietly dismantling the structures that life builds and returning their constituent elements to the environment where they can be used again. When a tree falls in a forest, it is fungi that begin the slow work of breaking it down, first colonizing the outer bark, then penetrating deeper into the sapwood and heartwood, digesting cellulose and lignin layer by layer until the trunk crumbles into soft, rich humus. When an animal dies, fungi join bacteria in the process of decomposition, breaking down proteins and fats, releasing nitrogen and phosphorus back into the soil. Leaves that fall in autumn are transformed by fungi over winter and spring, their tough cell walls digested, their nutrients liberated. Without this constant work of decomposition, nutrients would remain locked in dead tissue, unavailable to living organisms, and the cycles that sustain ecosystems would grind to a halt. Without fungi, dead organic matter would pile up endlessly across the earth. Imagine a world where nothing decayed, where every leaf that fell remained intact, where every tree that toppled lay unchanged for centuries, where the bodies of animals persisted indefinitely. Forests would become impenetrable tangles of dead wood. Grasslands would choke under layers of unrotted vegetation. Nutrients would become progressively locked away in inaccessible forms, and living plants would starve even as they stood atop mountains of organic matter. This is not a hypothetical scenario, but a glimpse of what Earth might have looked like during the Carboniferous period. Before fungi evolved, the enzymes needed to efficiently break down lignin, the structural polymer that gives wood its strength. During that era, dead trees accumulated faster than they decayed, eventually forming the vast coal deposits we mine today. It was only after fungi developed ligninase enzymes that the modern carbon cycle became possible, that forests could renew themselves, that nutrients could flow continuously from death back into life. Decomposition is not the opposite of life, but its necessary partner, the closing arc of a cycle that makes all growth possible. Fungi break down lignin, one of the toughest materials in plant cell walls. Lignin is the structural polymer that gives wood its remarkable strength and rigidity, the compound that allows trees to grow tall and support their massive weight against gravity. It is one of the most chemically resistant substances in nature, composed of complex aromatic rings bonded together in irregular three-dimensional networks that resist degradation. Most organisms cannot break these bonds. Lignin passes through digestive systems largely unchanged, accumulating wherever plant matter falls. But certain fungi, particularly the white rot Basidiomycetes, have evolved enzymes capable of cleaving lignin's stubborn bonds, oxidizing its aromatic structures and releasing the carbon trapped within. This ability is so rare and so essential that without it, dead wood would persist for centuries, forests would become graveyards of standing timber, and the global carbon cycle would collapse under the weight of undecomposed organic matter. Fungi are the only organisms on Earth that can efficiently return lignin to the atmosphere and soil, making them indispensable architects of nutrient flow in terrestrial ecosystems. Some fungi can decompose plastics in weeks instead of centuries. In recent years, researchers have discovered fungal species with the surprising ability to break down synthetic polymers, materials we designed specifically to resist degradation. Certain species of Aspergillus and Pestilentiopsis have been found colonizing plastic waste, secreting enzymes that cleave the carbon-carbon bonds in polyurethane, polyethylene, and other petroleum-based plastics. What normally takes hundreds of years to decompose through weathering and photodegradation can be reduced by these fungi in a matter of weeks or months. The enzymes work slowly but steadily, oxidizing the polymer chains, breaking them into smaller fragments, eventually into molecules simple enough to be absorbed and metabolized. It is a testament to the metabolic versatility of fungi, their capacity to adapt to novel substrates, to find energy in compounds that have existed on Earth for less than a century. In laboratories around the world, scientists are now studying these plastic-eating fungi, attempting to understand and harness their enzymatic pathways. Scientists are exploring fungi as a solution to the global plastic waste crisis. As plastic pollution accumulates in landfills and oceans, choking ecosystems and persisting for geological timescales, the search for biological solutions has intensified. Fungi offer a potential pathway, not a complete solution, but a promising tool in the broader effort to manage waste. Researchers are isolating and characterizing the enzymes that allow certain fungi to degrade plastics, working to optimize their activity through genetic modification and environmental manipulation. The goal is to develop industrial-scale bioreactors where plastic waste could be inoculated with fungal cultures and broken down into harmless byproducts or even into raw materials for manufacturing new products. There are challenges. The process is slower than mechanical recycling. The fungi are sensitive to temperature and moisture, and scaling up from laboratory to industrial application is never straightforward. But the principle has been proven. Fungi can digest what we once thought indigestible, can find nutrients in the detritus of industrial civilization. It is a small glimmer of hope in the face of an environmental crisis, a reminder that nature often holds solutions to problems of our own making. Certain fungi can break down toxic pollutants in contaminated soil. This process, known as mycoremediation, uses fungi to detoxify environments poisoned by industrial chemicals, petroleum products, pesticides, and other hazardous compounds. The same enzymatic systems that allow fungi to decompose lignin and cellulose can also degrade a wide range of synthetic toxins, breaking down the aromatic rings of petroleum hydrocarbons, cleaving the chlorinated bonds in pesticides, metabolizing compounds that are lethal to most other organisms. Fungi are introduced to contaminated sites, either by spreading spores or by inoculating the soil with mycelium grown in controlled conditions and allowed to colonize the substrate. Over weeks and months, the fungal network spreads through the contaminated zone, secreting enzymes, absorbing toxins, transforming dangerous compounds into benign metabolites. It is a slow, patient form of healing, operating on biological timescales, requiring careful monitoring and management, but in many cases proving more cost-effective and ecologically sound than excavation or chemical treatment. The fungi do not merely sequester the toxins, they break them down, genuinely neutralizing threats that would otherwise persist for decades. Some species can absorb and neutralize heavy metals. Unlike organic pollutants, heavy metals cannot be broken down. Lead, mercury, cadmium, and arsenic are elements, immutable in their atomic structure. But fungi have evolved mechanisms to sequester these toxic metals, binding them within their tissues and preventing them from leaching into groundwater or being taken up by plants. Certain fungal species accumulate heavy metals in concentrations hundreds or thousands of times greater than the surrounding soil, effectively mining them from contaminated sites. The metals bind to compounds in the fungal cell walls or are transported into vacuoles where they can be stored without harming the organism. Once the fungi have absorbed sufficient metals, they can be harvested and the contaminated biomass disposed of safely, often through high-temperature incineration that reduces the volume dramatically while concentrating the metals for potential recovery. It is a form of biological mining, using living organisms to extract elements from environments too toxic for conventional agriculture, gradually restoring land that industrial activity has rendered barren. Fungi were among the first organisms to colonize land, over 450 million years ago. In the Ordovician period, when Earth's continents were still bare rock and sand, devoid of soil or vegetation, fungi were already establishing themselves along the margins of ancient seas. Fossil evidence suggests they may have preceded plants onto land, or at the very least arrived simultaneously, pioneers venturing into an environment that had been sterile for billions of years. The transition from water to land was one of life's great challenges. Organisms had to adapt to desiccation, to ultraviolet radiation unfiltered by water, to temperatures that fluctuated wildly, to the mechanical stress of existing without the buoyant support of aquatic environments. Fungi, with their filamentous structure, their ability to grow through solid substrates, their capacity to form resistant spores, were pre-adapted to terrestrial life in ways that gave them an advantage. They spread across the bare rocks, beginning the slow work of breaking down mineral surfaces, creating the first primitive soils, preparing the ground for the plants that would follow. They helped plants make the transition from water to land by forming partnerships with algae. The earliest land plants, simple, rootless organisms that lacked the vascular tissues of modern species, could not have survived the harsh terrestrial environment alone. They formed symbiotic relationships with fungi, associations that may have been essential to their survival and success. Fossil evidence from 450 million-year-old rocks shows plant-like structures intimately associated with fungal filaments, suggesting that these partnerships are as old as terrestrial plant life itself. The fungi provided the plants with water and nutrients extracted from the primitive soil and weathered rock. The plants, capable of photosynthesis, provided the fungi with sugars. It was a mutualism born of necessity, two kingdoms supporting one another in an environment hostile to both. Some scientists believe that plants could not have colonized land without their fungal partners, that the greening of the continents depended on this ancient alliance forged in the Paleozoic seas and carried onto the bare shores of a waiting world. Mycorrhizal fungi form symbiotic relationships with about 90% of all plant species. This percentage is so high that it forces us to reconsider what we mean by a plant in the first place. The overwhelming majority of plants on Earth do not exist as independent organisms, but as composite beings, part plant, part fungus, their tissues intermingled at the cellular level in an intimacy so complete that neither partner can thrive without the other. These partnerships are called mycorrhizae, from the Greek words for fungus and root, and they represent one of the most successful collaborative strategies in the history of life. The association is ancient, dating back to the earliest terrestrial plants, and it has been maintained across hundreds of millions of years of evolution. Through mass extinctions and climate shifts, because it works, because both partners benefit, because together they can occupy niches that neither could survive alone. In forests, grasslands, tundra, and desert, beneath orchids and oaks and prairie grasses, the same fundamental partnership is being enacted. Fungal threads wrapped around or penetrating into root cells, trading minerals for sugar in an exchange as old as the soil itself. These fungi help plant roots access water and nutrients from the soil. A plant's roots, even when they branch extensively, can only explore a limited volume of soil. perhaps a few cubic meters for a large tree, far less for smaller plants. But mycorrhizal fungi extend far beyond the root zone, sending their filamentous hyphae outward in all directions, penetrating crevices too small for roots to enter, exploring a volume of soil dozens or even hundreds of times larger than the plant could access on its own. These fungal threads are far finer than the finest root hairs, allowing them to infiltrate tiny pores in the soil structure, where water and dissolved minerals accumulate. They are particularly efficient at scavenging phosphorus, an element essential for plant growth, but often locked in insoluble compounds that roots cannot absorb, and nitrogen in its various organic and inorganic forms. The hyphae also enhance water uptake, especially during periods of drought when moisture becomes scarce and the plant's own roots begin to dry out. Through the mycorrhizal connection, the plant gains access to resources it could never reach alone, a vast extension of its physiological reach accomplished not through its own tissues, but through a borrowed network woven by its fungal partner. In return, plants provide fungi with sugars created through photosynthesis. This is the other half of the exchange, the currency that flows in the opposite direction through the mycorrhizal connection. Plants, through the green alchemy of photosynthesis, convert sunlight, carbon dioxide, and water into glucose and other carbohydrates, energy-rich molecules that fungi, lacking chlorophyll, cannot produce. These sugars are transported from the leaves down through the phloem tissue of the stem and into the roots, where they are transferred across the cellular boundary into the waiting hyphae of the mycorrhizal fungus. Recent research suggests that plants may transfer more than 13 billion tons of carbon dioxide equivalent annually to mycorrhizal fungi worldwide, a staggering figure that represents roughly 36% of global fossil fuel emissions. This carbon becomes the fuel that powers the fungal network, allowing it to grow, to produce enzymes, to spread through the soil in search of nutrients. It is an exchange that benefits both partners profoundly. The plant gains access to mineral nutrients and water. The fungus gains access to carbohydrates it cannot manufacture. And because each partner invests in the other's success, because the fungus prospers when its host plants thrive, and the plants flourish when their fungal networks are robust, the relationship is self-reinforcing, stable across evolutionary time. Some plants cannot survive without their fungal partners. For these species, the mycorrhizal association is not optional or merely beneficial, but obligate, a relationship without which germination fails, growth stalls, and death follows. Many tree species, particularly those in nutrient-poor soils or harsh climates, depend absolutely on mycorrhizal fungi for establishment and survival. Without their fungal partners, seedlings struggle to absorb sufficient phosphorus and nitrogen. Their growth becomes stunted, their leaves turn pale and chlorotic, and they eventually succumb to nutrient deficiency, even in soil that appears rich in organic matter. The dependency is so complete that foresters attempting to establish plantations in areas lacking appropriate mycorrhizal fungi must inoculate the soil or treat seedlings with fungal cultures before transplanting, or the trees will simply fail to thrive. This obligate relationship reveals how deeply intertwined the lives of plants and fungi have become over hundreds of millions of years of coevolution. How what began as a facultative partnership in some ancient ancestor has, in many lineages, become an essential fusion, a biological integration so complete that neither organism can exist independently. Orchids depend entirely on specific fungi to germinate their seeds. orchid seeds are among the tiniest in the plant kingdom. Mere specks of tissue containing an embryo, but almost no stored food reserves, no endosperm to fuel early growth. A single orchid capsule may contain tens of thousands or even millions of these dust-like seeds, scattered on the wind in the hope that a few will land in suitable habitat. But suitable habitat for an orchid seed means soil that contains the right species of mycorrhizal fungus, because without fungal assistance, the seed cannot germinate. The fungus infects the seed shortly after it lands, penetrating the embryo and providing it with carbohydrates and nutrients absorbed from decaying organic matter in the soil. The embryo, lacking chlorophyll and unable to photosynthesize, depends entirely on this fungal subsidy during its first months or even years of life. Some orchid species remain underground for several years as protocorms, swollen embryonic structures nourished entirely by their fungal partners, before finally sending up their first green leaves and beginning photosynthesis. Even after maturity, many orchids maintain their mycorrhizal relationships, continuing to receive fungal support throughout their lives. This absolute dependency explains why orchids are so difficult to cultivate and why they are so sensitive to habitat disturbance. They require not only the right soil, moisture, and light conditions for themselves, but also the presence of specific fungal species without which they cannot even begin their lives. Fungal spores are incredibly resilient and can survive extreme conditions. Spores are the fungal equivalent of seeds, microscopic packets of genetic material encased in protective walls, designed to endure harsh environments and disperse to new locations. But whereas seeds contain embryos and stored nutrients, spores are simpler, often single-celled structures that can remain dormant for extended periods, waiting for conditions favorable enough to germinate. The spore walls are constructed from layers of tough, chemically resistant materials that protect the genetic contents from desiccation, ultraviolet radiation, temperature extremes, and chemical assault. Some spores can survive being frozen in Arctic permafrost for decades. Others endure the searing heat of desert soils or the caustic conditions of industrial waste sites. Their resilience allows fungi to colonize environments that would be lethal to actively growing organisms, to bridge gaps in time and space that would doom less hardy structures. When a spore finally encounters suitable conditions, adequate moisture, appropriate temperature, a substrate rich in nutrients, it swells, the wall cracks open, and a hyphal thread emerges, beginning the process of growth and colonization all over again. It is a strategy of patience and endurance, of waiting out adversity in a state of suspended animation until opportunity returns. Mushroom spores have been shown to survive the vacuum and radiation of space. In experiments where spores were exposed to the harsh conditions beyond Earth's atmosphere, absolute vacuum, temperatures near absolute zero, intense ultraviolet and cosmic radiation unfiltered by any atmosphere, a significant proportion remained viable. When returned to favorable conditions and provided with moisture and nutrients, these space-exposed spores germinated and grew normally, as if their journey through the void had been merely a pause in their life cycle, rather than a brush with annihilation. This extraordinary resilience suggests that fungal spores are among the most durable biological structures known, capable of withstanding conditions that would instantly destroy most living cells. It also raises intriguing questions about panspermia, the hypothesis that life might travel between worlds on meteorites and comets. Because if spores can survive the vacuum of space, perhaps they could also survive ejection from a planet's surface during an asteroid impact, drift through space for millennia, and seed life on distant worlds. Whether fungi have actually made such journeys remains unknown, but their capacity to do so has been demonstrated in laboratories orbiting Earth. Spores can remain dormant for years, waiting for the right conditions to grow, unlike seeds, which typically remain viable for months, or at most a few years before their stored nutrients degrade and their embryos die. Fungal spores can persist in a state of suspended animation for decades or longer. They achieve this longevity through a combination of extreme desiccation, reducing their water content to nearly zero, and the construction of multi-layered cell walls that protect the genetic material and cellular machinery from oxidative damage. In this dormant state, metabolic processes slow to near imperceptible levels. The spore is alive, but barely, consuming no resources, generating no waste, simply waiting. When conditions finally become favorable, when moisture returns, when temperature rises or falls into the appropriate range, when substrates rich in nutrients become available, the spore absorbs water, swells, and germinates, sending out a delicate hyphal thread to begin the cycle of growth anew. This ability to wait, to endure inhospitable periods without growth or reproduction, allows fungi to colonize environments characterized by long periods of unsuitability, punctuated by brief windows of opportunity, deserts that bloom after rare rains, Arctic soils that thaw for only a few weeks each year, disturbed habitats where succession unfolds slowly over decades. A single mushroom can release billions of spores into the air. The numbers are almost incomprehensible. A large puffball or bracket fungus may produce trillions of spores over its lifespan, releasing them in clouds so dense they become visible as smoke-like plumes drifting on the wind. Even modest-sized mushrooms release millions or tens of millions of spores during their brief reproductive period, usually just a few days when conditions are optimal. These spores are produced on specialized structures called basidia or asci, microscopic cells that line the gills or pores of the mushroom's underside, each producing 4 or 8 spores before dying and being replaced. The sheer abundance reflects the precariousness of fungal dispersal. Of the billions of spores released, only a tiny fraction will land in locations suitable for germination, and of those that germinate, fewer still will establish successful mycelia, capable of producing their own fruiting bodies. It is a strategy of overwhelming abundance, of compensating for enormous mortality through sheer numbers, scattering genetic material across the landscape in the hope that a few propagulos will find purchase and continue the lineage. Spores are so light they can travel thousands of miles on the wind. The average fungal spore measures between 2 and 20 micrometers in diameter, smaller than a red blood cell, light enough to float on the slightest air current. Once released into the atmosphere, spores can remain airborne for hours or days, carried by wind currents to altitudes of several kilometers, where they join the aeroplankton, the invisible community of microorganisms, pollen grains, and dust particles suspended in the air we breathe. Atmospheric circulation can transport these spores across continents and oceans, from one hemisphere to another, dispersing fungal species far beyond the range of any animal-mediated dispersal mechanism. This long-distance dispersal explains why many fungal species have cosmopolitan distributions, appearing on multiple continents despite having no obvious means of crossing oceanic barriers. A spore released from a mushroom in a European forest might drift across the Atlantic and germinate in a North American woodland weeks later, establishing a new population thousands of miles from its origin. It is a form of migration so gradual and so diffuse that it operates on timescales beyond human perception. Yet over geological time, it has shaped the global distribution of fungal diversity. Some fungi create their own airflow to help disperse their spores more effectively. Certain mushroom species, particularly those that release spores from gills or pores, generate convective currents through the process of water evaporation from their surfaces. As water evaporates from the mushroom's moist tissue, it cools the surrounding air, creating a temperature gradient that induces gentle upward airflow. This rising air carries spores away from the mushroom surface and lifts them above the boundary layer, the zone of still air that clings to surfaces and would otherwise trap spores near the ground where they might simply fall back onto the forest floor. Other species use more active mechanisms. Some puffballs forcibly eject their spores through small openings at the top of the fruiting body when raindrops strike their outer surface, using the mechanical impact to create pressure waves that propel spores into the air. A few species even generate electrical fields that repel spores from the gill surface, overcoming the adhesive forces that would otherwise keep spores clinging to the structures that produce them. These self-dispersal mechanisms, though operating on microscopic scales, significantly enhance reproductive success by ensuring that spores enter air currents capable of carrying them to new locations rather than simply falling into the immediate vicinity of the parent organism. Certain mushrooms glow softly in the dark through a process called bioluminescence. In tropical and subtropical forests around the world, scattered among the leaf litter and growing on rotting logs, are mushrooms that emit a faint greenish light visible in complete darkness. This ghostly glow, called foxfire in folklore, results from a chemical reaction in which an enzyme called luciferase catalyzes the oxidation of a compound called luciferin, releasing energy in the form of photons. The same basic biochemistry powers the bioluminescence of fireflies, deep-sea fish, and certain bacteria, though each lineage has evolved the trait independently, converging on similar molecular solutions to the challenge of producing light without heat. In fungi, bioluminescence appears to have evolved multiple times in distantly related lineages, suggesting that the ability confers some selective advantage, though what that advantage might be remains a subject of scientific debate. The light is dim, typically barely bright enough to read by, but in the absolute darkness of a moonless forest night, even this faint glow becomes visible, creating constellations of light scattered across the forest floor like terrestrial stars. Over 80 species of bioluminescent fungi have been identified. These species belong to several different families and genera, widely distributed across tropical and temperate regions, though most luminous species are found in warm, humid environments where fungi flourish year-round. Some of the best-known bioluminescent species include Pinellus stypticus, which grows on decaying hardwood in North America and Europe, and various species of Mycena found in Asian and South American rainforests. Omphallotus olearius, the jack-o'-lantern mushroom, produces a particularly striking bioluminescence, visible even in dim twilight. Each species has its own characteristic light intensity and temporal pattern. Some glow continuously throughout the night, while others pulse or flicker at regular intervals. The diversity of bioluminescent fungi suggests that this trait, while uncommon relative to the total number of fungal species, has proven successful enough to be maintained across millions of years of evolution, spreading through various fungal lineages and persisting in modern species. These glowing mushrooms emit a greenish light that can be seen at night in forests. The wavelength of fungal bioluminescence typically peaks around 520–530 nanometers in the green portion of the visible spectrum, the same color produced by fireflies and many other bioluminescent organisms. This particular wavelength is no accident. Green light travels through forest air with minimal scattering and absorption, making it visible at greater distances than other colors would be. On humid nights when the air is still and darkness is complete, these glowing mushrooms become faintly visible from several meters away, their soft radiance creating an ethereal atmosphere in forests lucky enough to harbor them. The intensity varies with environmental conditions. Fresh, actively growing specimens produce brighter light than older, deteriorating ones, and the glow intensifies in warm, humid conditions when fungal metabolism runs fastest. In some locations where bioluminescent species are abundant, the cumulative effect of dozens or hundreds of glowing mushrooms scattered across the forest floor creates a subtle illumination, a gentle radiance that transforms the nighttime forest into something magical and otherworldly. The glow may attract insects that help spread the fungal spores. This hypothesis that bioluminescence serves as a form of advertisement to attract spore-dispersing insects remains debated among mycologists, but experimental evidence supports it. Studies have shown that certain insects, particularly beetles and flies, are attracted to bioluminescent fungi and spend more time on or near glowing mushrooms than on non-luminous specimens. If these insects subsequently visit other mushrooms or move through the forest with spores adhering to their bodies, they could function as dispersal vectors, carrying fungal spores to new locations more effectively than wind alone. This would make fungal bioluminescence analogous to the bright colors and fragrances of flowers, signals designed to attract animal partners that provide reproductive services in exchange for incidental benefits. The relationship would not be as tightly co-evolved as plant pollinator mutualisms, since the insects likely gain no nutritional reward from visiting luminous mushrooms, but even passive attraction could enhance dispersal enough to favor the maintenance of bioluminescence across evolutionary time. Bioluminescent fungi have been used as natural lanterns in some cultures. In remote tropical regions where electric light was unknown and candles were scarce, people would collect glowing mushrooms and place them along forest paths or carry them as gentle sources of illumination during nighttime journeys. The light was too dim for reading or fine work, but sufficient to navigate by, to see the outline of a trail, to distinguish roots from shadows. Travelers would arrange these luminous mushrooms in small clusters, creating waymarkers that glowed with ghostly green radiance through the darkness. It is a reminder that long before we learned to capture fire in glass bulbs or generate electricity from distant power stations, nature had already solved the problem of light in darkness through quiet chemistry, through the patient work of enzymes transforming molecules into photons. Fungi come in an astonishing array of colors, shapes, and sizes. From the delicate translucent caps of mycelial species that seem barely substantial enough to exist, to the massive shelf-like brackets of Ganoderma that jut from tree trunks like architectural features, from tiny pinmold colonies invisible to the naked eye, to giant puffballs the size of soccer balls, the fungal kingdom encompasses a diversity of form that rivals any other group of organisms. Each shape represents a different strategy for survival, a different relationship with decay and dispersal, a different solution to the problem of how to live by consuming what has already died. Some fungi are flat and encrusting. spreading across bark like living paint. Others rise on slender stalks as delicate as glass. Still others form intricate branching structures that resemble coral or antlers or the frozen splash of water. The variety seems endless, each form sculpted by millions of years of evolution into its particular ecological niche, each one a small masterpiece of biological engineering. Some are smaller than a pinhead, others spread across miles of forest. The smallest fungi are microscopic yeasts, single cells floating in liquid, barely visible even under magnification. Entire organisms contained within a space measured in micrometers. The largest, like the Oregon humongous fungus we encountered earlier, sprawl across thousands of acres, their mycelial networks threading through soil and wood in a vast invisible web that connects entire ecosystems. Between these extremes lies every imaginable gradation of size and scale, from molds that colonize a single fallen leaf to mushrooms that weigh several pounds. It is humbling to realize that the same kingdom contains organisms separated by 8 or 9 orders of magnitude in physical extent, yet all operating according to the same fundamental principles of fungal biology, the same reliance on external digestion, the same branching mycelial architecture, the same patient relationship with time. The color of a mushroom can range from pure white to deep purple, bright red or electric blue. Some species glow with pigments so vivid they seem almost artificial. The scarlet caps of Amanita muscaria dotted with white spots like something from a fairy tale. The indigo gills of certain Cortinaria species that stain your fingers blue when touched. The sulfur yellow flesh of chicken of the woods mushrooms that cascade down tree trunks in brilliant tears. These colors serve various functions, some warn of toxicity, advertising danger to would-be consumers. Others attract spore-dispersing animals with promises of food. Still others simply result from the chemistry of pigment production, with no apparent adaptive value. Accidents of metabolism that happen to be beautiful. Walking through a woodland in autumn, you might encounter white, tan, orange, pink, purple, green, and black mushrooms, all within a few square meters, a palette as diverse as any flower garden. Each hue speaking to a different chemistry, a different way of being. Fungi produce some of the most potent natural chemicals known to science. The chemical arsenal of fungi includes compounds that kill bacteria, suppress immune systems, alter consciousness, induce visions, stop hearts, and cure diseases. Many of these substances evolved as defensive chemicals, ways for fungi to protect their territories from competitors or predators, to fend off bacterial invasion, to secure their patch of substrate in a world where everything decays and nothing belongs to anyone for long. But humans have learned to harness these compounds for medical and industrial purposes, recognizing that the same molecule that a fungus uses to wage microscopic warfare in a rotting log can, when purified and administered carefully, save a human life. It is a testament to the sophistication of fungal biochemistry, to the hundreds of millions of years these organisms have spent perfecting the art of chemical synthesis. Penicillin, one of the world's first antibiotics, comes from a fungus. In 1928, Alexander Fleming returned from vacation to his laboratory at St. Mary's Hospital in London to find that a penicillium mold had contaminated one of his bacterial cultures, and around the mold, the bacteria had died. A clear zone of inhibition surrounding the fungal colony like a protective barrier. This accidental observation, born of what might have been dismissed as mere sloppiness, led to the isolation of penicillin, a compound that would transform medicine and alter the course of human history. It took more than a decade to purify penicillin in sufficient quantities for medical use, but once it became available, it proved devastatingly effective against bacterial infections. It was a fungus, a common mold of the sort that grows on forgotten bread, that appears unbidden on old fruit, that provided humanity with its first powerful weapon against bacterial infection. That opened the door to the age of antibiotics. This discovery has saved countless millions of lives since the 1940s. Before antibiotics, simple infections could prove fatal. A cut on the hand might lead to sepsis and death. Pneumonia was a leading cause of mortality. Childbirth fever killed mothers with terrifying regularity. Penicillin changed everything, allowing doctors to treat pneumonia, strep throat, syphilis, wound infections, and countless other bacterial diseases that had previously carried significant mortality. It is estimated that antibiotics have extended average human lifespan by more than a decade, and penicillin was the first and remains among the most important. A gift from the fungal world that continues to save lives every day, in hospitals and clinics across the globe. A reminder that some of our most profound medical advances came not from human ingenuity alone, but from careful observation of what nature had already invented. Many other life-saving medicines are derived from fungi. Beyond penicillin, fungi have given us cyclosporin, used to prevent organ rejection in transplant patients, and statins, which lower cholesterol and reduce heart disease risk. They produce compounds used to treat migraines, to manage blood pressure, to fight certain cancers. Researchers continue to screen fungal species for new pharmaceutical compounds, recognizing that the chemical diversity of the fungal kingdom represents a largely untapped reservoir of potential medicines. Each fungal species synthesizes its own unique suite of secondary metabolites, and with millions of species yet to be discovered, the possibility remains that fungi hold cures for diseases we do not yet know how to treat. Some fungi are used to create immunosuppressants for organ transplant patients. Cyclosporin, discovered in a soil fungus collected from Norway, revolutionized transplant medicine by allowing doctors to suppress the immune system just enough to prevent organ rejection without leaving patients defensively vulnerable to every passing infection. Before this fungal compound became available, organ transplants had far lower success rates, with many patients' bodies rejecting the foreign tissue despite aggressive treatment. Now, thanks to this molecule refined from fungal metabolism, transplant patients can live for decades with donated organs, their bodies accepting foreign tissue as if it were their own. Others produce statins, which are used to lower cholesterol. These compounds, first isolated from Penicillium and Aspergillus species, inhibit an enzyme involved in cholesterol synthesis, reducing blood cholesterol levels and thereby lowering the risk of heart attack and stroke. Millions of people worldwide take statin medications daily, most never knowing that the molecular structure they depend on was originally designed by a fungus as a secondary metabolite, as chemical byproduct of some ancient adaptation to life in the soil. Fungi are also used in traditional medicine systems around the world. In Chinese medicine, various mushroom species have been prescribed for thousands of years to treat ailments ranging from respiratory infections to cancer, from fatigue to immune deficiency. Indigenous cultures across continents have recognized the medicinal properties of local fungal species, developing sophisticated knowledge systems about which species heal and which harm, which restore vitality and which bring visions. This traditional knowledge, accumulated over centuries of careful observation, is now being investigated by modern science, which is discovering that many of these folk remedies contain compounds with genuine pharmacological activity. Certain mushrooms have been used for thousands of years in healing rituals. Reishi mushrooms were revered in ancient China as the mushroom of immortality, prescribed for longevity and vitality, depicted in art and literature as symbols of health and spiritual attainment. Traditional healers in Siberia, Mesoamerica, and Africa have long used specific fungal species in ceremonies designed to treat both physical and spiritual ailments, recognizing that health encompasses more than the mere absence of disease. These practices persist today, carrying forward knowledge that predates written history. Reishi, lion's mane, and cordyceps are valued for their medicinal properties. Modern research has begun investigating the compounds in these traditional medicines, finding substances that may indeed support immune function, cognitive health, and energy metabolism. Lion's mane contains compounds that appear to stimulate nerve growth factor. Cordyceps may enhance athletic performance and cellular energy production. Reishi contains polysaccharides that modulate immune response. Whether these fungi will prove as beneficial as traditional knowledge suggests remains an active area of scientific study, but their centuries-long use across multiple cultures speaks to observed effects that merit serious investigation. At least 216 species of fungi are known to have hallucinogenic effects. This number represents our current knowledge, the species that have been tested, documented, and confirmed to contain psilocybin, psilocin, or related psychoactive compounds. The actual number may be higher, as many fungal species remain unstudied, their chemical profiles unknown. These hallucinogenic fungi are distributed across several genera, including psilocybe, Paniolus, Gymnopilus, Pluteus, and others, each with their own characteristic appearance, habitat preferences, and potency. They grow on every continent except Antarctica, though they are most diverse in tropical and subtropical regions, where warm, humid conditions favor fungal growth year-round. It is a remarkable chemical convergence, different lineages independently evolving the capacity to produce compounds that profoundly alter human consciousness. Psilocybin mushrooms have been used in spiritual ceremonies for millennia. Archaeological evidence and historical records suggest that indigenous cultures in Mesoamerica were consuming these mushrooms in religious contexts long before European contact. The Aztecs called them Teonanacatl, flesh of the gods, and used them in rituals designed to communicate with the divine, to receive visions, to heal spiritual ailments. Similar practices existed in other parts of the world, though the historical record is less complete. These ceremonies were not recreational but sacramental, conducted with reverence and ritual structure, guided by experienced practitioners who understood the power of the substances they administered. The mushrooms were treated as medicines for the soul, as tools for accessing hidden dimensions of experience. Modern research is exploring their potential to treat depression and anxiety. After decades of prohibition that halted scientific investigation, psilocybin research has resumed, and early results have been striking. Clinical trials have shown that a single high dose of psilocybin, administered in a controlled setting with psychological support, can produce lasting reductions in depression and anxiety, particularly in patients with treatment-resistant conditions or those facing terminal illness. The mechanism appears to involve a temporary disruption of rigid thought patterns, allowing patients to experience their lives and challenges from new perspectives. It is too early to declare psilocybin a miracle cure, but the preliminary evidence suggests that these ancient fungal compounds may have legitimate therapeutic applications in modern psychiatry. Fungi have been used to produce alcoholic beverages for at least 9,000 years. Archaeological evidence from ancient China reveals that people were fermenting rice, honey, and fruit into alcoholic drinks as early as 7000 BCE. Similar evidence exists from the Middle East and elsewhere, suggesting that humans discovered fermentation independently in multiple locations as agriculture developed. These early fermentations were likely initiated by wild yeasts present in the environment or on the raw materials themselves. People did not understand what caused the transformation, the bubbling, the sweetness, turning to intoxication, but they recognized it as valuable, as something to be preserved and repeated. Fermentation became one of humanity's first biotechnologies, practiced for thousands of years before anyone knew that microorganisms existed. Yeasts, a type of single-celled fungus, are essential to brewing and baking. Unlike the filamentous fungi that form visible mycelia and mushrooms, yeasts live as individual cells floating in liquid or dusting the surfaces of fruit and grain. They are still fungi, still members of the same kingdom, but simplified, miniaturized, adapted to aquatic environments. When yeast consume sugar in the absence of oxygen, they produce alcohol and carbon dioxide as waste products, a metabolic pathway called fermentation that humans have harnessed for brewing beer, fermenting wine, and leavening bread. Without yeast, there would be no beer, no wine, no bread as we know it. These microscopic fungi have shaped human culture and cuisine for millennia, long before anyone understood their nature. Without yeast, there would be no bread, beer, or wine. Imagine human history without these staples, without the leavened loaves that allowed civilizations to develop portable, shelf-stable food, without the fermented beverages that made contaminated water safe to drink and provided calories during lean seasons. Brewing and baking are so fundamental to human culture that they appear in our oldest written records, in religious texts and legal codes, in art and literature. And all of it depends on the metabolic activities of yeasts, on their capacity to convert sugar into alcohol and gas, on chemistry that occurs inside cells too small to see. We have been in partnership with these fungi for thousands of years, though for most of that time we did not know our partners existed. Fungi are also responsible for fermenting foods like soy sauce, miso, and tempeh. In East and Southeast Asian cuisines, filamentous fungi, particularly species of Aspergillus and Rhizopus, are used to transform soybeans and grains into foods with complex flavors and improved digestibility. Soy sauce begins with Aspergillus fungi breaking down proteins and starches in soybeans and wheat, creating amino acids and sugars that are then fermented further by bacteria. Tempeh is made by inoculating cooked soybeans with Rhizopus oligosporus, which grows a white mycelial mat that binds the beans together into a firm cake rich in protein and vitamins. Miso involves a similar fungal fermentation, followed by bacterial aging. These foods have sustained populations for centuries, providing essential nutrients in regions where other protein sources were scarce. Cheese depends on fungi for its texture, flavor, and preservation. While bacteria play the primary role in converting milk to cheese, fungi contribute significantly to the character of many varieties. Molds growing on cheese surfaces or within their interiors produce enzymes that break down fats and proteins, creating the complex flavors and aromas that distinguish one cheese from another. Some cheeses are intentionally inoculated with specific fungal species. Others acquire their molds from the environment of the aging caves where they mature. The relationship between cheese and fungi is ancient, discovered through trial and error in the centuries before refrigeration, when preserved foods meant survival through winter. Blue cheese gets its distinctive veins from a species of penicillium mold. The same genus that gave us penicillin also gave us Roquefort, Gorgonzola, and Stilton. Penicillium roqueforti is introduced into the cheese during production, and as it grows through the interior, it forms the characteristic blue-green veins and produces compounds that give these cheeses their sharp, pungent flavors. The mold requires oxygen to grow, so cheesemakers pierce the aging wheels with long needles to create air channels. What might appear as spoilage is actually careful cultivation, a controlled fungal infection that transforms bland curds into something prized and complex. At least 350 species of fungi are eaten as food around the world. From the delicate enoki mushrooms of Japanese cuisine to the meaty portobellos grilled in Mediterranean kitchens, from the earthy porcini that flavor Italian risottos to the prized matsutake of Korea and Japan, edible mushrooms appear in culinary traditions across every inhabited continent. Each species brings its own texture, flavor, and nutritional profile. Some nutty, some fruity, some reminiscent of meat or seafood. Gathering wild mushrooms remains a cherished tradition in many cultures, a seasonal ritual that connects people to the forest and to knowledge passed down through generations about which species are safe, which are choice, and which must be left untouched. Truffles, one of the most expensive foods on Earth, are underground fungi. Unlike mushrooms that push up through soil to release their spores into the air, truffles form entirely beneath the surface, often several inches down, in intimate association with the roots of specific tree species, oaks, hazels, poplars. They rely on animals to find them, dispersing their spores when wild boars, deer, or rodents dig them up, attracted by the powerful aroma the truffles emit when ripe. This subterranean lifestyle and the difficulty of cultivation make truffles exceedingly rare and correspondingly valuable. Trained dogs and pigs are used to locate them in European woodlands, where they grow, sniffing out the distinctive scent that humans cannot detect until the truffle is unearthed. A single white truffle can sell for thousands of dollars. The white truffle of Alba, Italy, Tuber magnatum, is among the rarest and most prized, with exceptional specimens at auction fetching prices that rival precious metals. Their season is brief, their habitat specific, and they cannot yet be reliably cultivated. What you pay for is scarcity, certainly, but also a flavor and aroma so intense and complex that a few shavings can transform a simple dish into something transcendent. It is perhaps the clearest example of how fungi, these humble decomposers, can become objects of luxury and desire. Mushrooms are a rich source of vitamins, minerals, and protein. Despite consisting of about 90% water, mushrooms pack considerable nutrition into their remaining mass: B vitamins, selenium, copper, potassium, and in some species, significant amounts of vitamin D when exposed to sunlight. They provide protein and fiber while remaining low in calories and fat. Many species contain bioactive compounds, polysaccharides, terpenoids, phenolic compounds that appear to offer health benefits beyond basic nutrition. For populations where meat is scarce or avoided, Mushrooms have long served as an important protein source and flavor enhancer. Some fungi are deadly poisonous, containing toxins that can destroy the liver or kidneys. These are not irritants or mild toxins that cause temporary discomfort, but compounds of devastating potency, amatoxins that bind to enzymes essential for protein synthesis, shutting down cellular function in the liver, kidneys, and other organs. Symptoms may not appear for hours or even a day after ingestion, by which time the toxins have already begun their work. Treatment is supportive. There is no antidote, only the hope that the body can survive long enough to regenerate damaged tissues. The death rate from amatoxin poisoning remains tragically high, even with modern medical care. The death cap mushroom is responsible for most fatal mushroom poisonings worldwide. Amanita phalloides, innocent in appearance, a pale mushroom with white gills and a delicate veil, contains enough amatoxins in a single cap to kill an adult. It is widely distributed across Europe, Asia, and increasingly in North America, and other regions where it has been accidentally introduced. The death cap often grows near oaks, appearing in late summer and autumn, when other choice edible mushrooms are also fruiting, increasing the risk of mistaken identity. Experienced foragers treat Amanita species with extreme caution, learning to recognize the distinctive features, the sac-like vulva at the base, the ring on the stem, that mark them as dangerous. Many poisonous mushrooms closely resemble edible species. This mimicry, though not intentional, creates genuine peril for those who gather wild mushrooms without expertise. The death cap can be confused with edible agaricus species. Toxic Galerina mushrooms grow on wood, alongside choice edibles, and share similar coloring. Field guides emphasize that there is no simple rule, no test of silver spoons or garlic cloves, no reliable shortcut that can distinguish safe from dangerous. The only protection is knowledge, learning to identify species with certainty, understanding habitat and season, examining every specimen carefully. It is a reminder that the fungal kingdom, for all its gifts, demands respect and caution in equal measure. Fungi exhibit a staggering diversity of survival strategies. Over hundreds of millions of years, they have adapted to nearly every conceivable ecological niche, from the Arctic tundra to tropical rainforests, from desert soils to the deep ocean, from pristine wilderness to contaminated industrial sites. Some are generalists, capable of decomposing a wide range of organic matter. Others are specialists so refined in their requirements that they can grow only on a single host species or substrate. There are parasites that kill their hosts, mutualists that enhance them, and saprobes that wait patiently for death before beginning their work. This ecological diversity reflects the fundamental versatility of the fungal body plan, a network of threads that can grow through solid substrates, secreting enzymes and absorbing nutrients, adaptable to countless variations in strategy and circumstance. Cordyceps fungi infect insects, taking over their bodies and minds. These are not simple parasites, but organisms that manipulate host behavior in ways that seem almost impossible. A fungal spore lands on an ant or beetle. It germinates, penetrates the exoskeleton, and begins growing through the insect's body. But rather than killing the host immediately, the fungus keeps it alive, its mycelium spreading through tissues, eventually reaching the brain. The infected insect begins behaving strangely, abandoning its colony, climbing vegetation at specific times of day, positioning itself in locations that optimize fungal reproduction. An infected ant will climb to a high point before the fungus sprouts from its head. This is the endpoint of the infection. The ant, compelled by chemical signals from the fungus, climbs to a location elevated above the forest floor, clamps its jaws onto a leaf or twig in a death grip, and dies. Hours or days later, a fruiting body emerges from the back of the ant's head, growing upward and eventually releasing spores that drift down onto the forest floor below, where other ants pass and potentially become infected. The precision of this behavior, the timing, the height, the orientation, suggests that the fungus has evolved mechanisms to control specific neural circuits in its host, though how this is accomplished remains incompletely understood. This behavior helps the fungus spread its spores over a wider area. By compelling its host to climb before dying, the fungus achieves what it cannot do on its own: elevation. Spores released from ground level would settle nearby, limiting dispersal. Spores released from even a few centimeters of height can travel much farther on air currents. It is a strategy that maximizes reproductive success through behavioral manipulation, a testament to the evolutionary arms race between pathogens and hosts. And while it seems macabre from a human perspective, it is simply another variation on the theme of survival, another way that fungi have learned to persist and propagate in a competitive world. Oyster mushrooms are carnivorous, trapping and digesting tiny nematodes. This fact surprises many people who think of mushrooms as passive decomposers of dead matter. But several species of oyster mushrooms, including Pleurotus austriatus, actively hunt microscopic worms called nematodes that live in soil and rotting wood. The fungus produces specialized structures, sticky knobs or constricting loops, that trap passing nematodes, immobilizing them. Once caught, the fungal hyphae penetrate the nematode's body and secrete enzymes that digest it from the inside, absorbing the nitrogen-rich proteins that nematodes contain. This carnivorous behavior likely evolved as an adaptation to nitrogen-poor environments, allowing the fungus to supplement its diet with animal protein when plant matter alone proves insufficient. Some fungi form fairy rings, circular patterns that can expand for centuries. These mysterious circles appear in grasslands and forests worldwide, rings of mushrooms, or zones where the grass grows darker and more lush, or sometimes where vegetation dies back entirely. They form when a fungal spore germinates and begins growing outward in all directions equally, creating a circular mycelial network beneath the surface. Each year, the fungus extends a bit farther from the center, and when conditions are right, mushrooms fruit along the expanding perimeter. Some fairy rings are ancient, centuries old, their diameters measuring dozens of meters, silent testimony to patient, incremental growth sustained across generations. These rings grow outward as the mycelium exhausts nutrients at the center. As the fungus consumes available organic matter in the soil, it must expand into fresh territory to find new food sources. The center of the ring becomes depleted, the mycelium there dying back or becoming dormant, while the leading edge continues advancing outward in an ever-widening circle. Growth rates vary by species and environment, but typically range from a few centimeters to half a meter per year. By measuring the diameter of a fairy ring and estimating the growth rate, scientists can approximate its age, and some rings appear to be 600 years old or more, predating the oldest trees in the forests where they grow. Fairy rings have inspired myths and folklore across many cultures. In European tradition, these circles were said to mark places where fairies danced at night, or doorways to the fairy realm where mortals might vanish if they stepped inside. Dutch and German folklore warned that fairy rings were caused by the devil churning butter, while Scandinavian tales spoke of elves gathering in these enchanted circles. The appearance of mushrooms overnight, their perfect circular arrangement, their persistence in the same location year after year. All of this seemed magical to people who did not know about mycelial networks growing beneath the soil. Even now, encountering a fairy ring in a misty meadow, at dawn carries a sense of quiet wonder, a reminder that not all mysteries require supernatural explanation to feel enchanted. Fungi can survive in some of the harshest environments on Earth. While we often picture fungi in moist, temperate forests, they have colonized nearly every habitat where life exists at all, from deserts where rain falls once a year to hot springs, where temperatures approach boiling, from hypersaline lakes to acidic mine tailings. Their filamentous structure, their ability to produce resilient spores, and their versatile metabolism allow them to persist where other organisms cannot. Some species endure by growing slowly during brief favorable periods and remaining dormant the rest of the time. Others have evolved specialized adaptations that let them thrive in conditions that would kill most living things. They have been found in the frozen depths of Antarctica, in rock crevices and beneath the surface of glacial ice, in cryptoendolithic communities where they grow inside porous stones. Fungi survive temperatures that rarely rise above freezing. They metabolize at rates so slow they are barely measurable, growing perhaps a few cells per year, but they persist. Antarctic fungi have adapted to extreme cold, to intense ultraviolet radiation, to desiccation and nutrient scarcity. Their presence in these frozen wastes suggests that life, given even the slightest foothold, will find ways to endure in places that seem utterly inhospitable. Some fungi thrive in the high radiation environment of Chernobyl's reactor. After the 1986 nuclear disaster, the ruins of Reactor 4 became one of the most radioactive places on Earth, lethal to humans, abandoned to decay. Yet within a few years, scientists noticed black fungi growing on the reactor walls, thriving in radiation levels that would be instantly fatal to most organisms. These melanized fungi were not merely surviving, but appeared to be growing toward radiation sources, colonizing the most contaminated areas preferentially. These fungi use melanin to convert radiation into chemical energy. The same pigment that colors human skin and protects us from sunlight appears to serve an entirely different function in these fungi, acting as a kind of biological solar panel for ionizing radiation. The melanin absorbs gamma rays and somehow channels that energy into chemical processes the fungus can use for growth, analogous to how chlorophyll captures light energy in photosynthesis. The mechanism is still being studied, but the implications are profound. Life finding a way not just to tolerate, but to harness one of the most destructive forces we know. Researchers are studying whether fungi could protect astronauts from cosmic radiation. Space beyond Earth's protective magnetic field is awash in radiation. Solar particles, cosmic rays, energetic enough to damage DNA and increase cancer risk during long missions. Current shielding is heavy and expensive to launch. Biological solutions might offer an alternative. If melanized fungi can be grown as living radiation shields on spacecraft or habitats, they could reduce exposure while requiring minimal resources. It remains speculative, but experiments aboard the International Space Station have already tested how these fungi perform in microgravity. Fungi could one day help terraform other planets by breaking down rock into soil. On Earth, lichens, symbiotic partnerships between fungi and algae, are among the first colonizers of bare rock, secreting acids that slowly weather stone into mineral particles. Over centuries, this biological weathering creates the substrate in which other organisms can eventually grow. On Mars or other rocky worlds, fungi might play a similar pioneering role, beginning the long process of transforming sterile regolith into something approaching soil. It is a vision of deep time and patient transformation, of life spreading beyond its birth world through the quiet work of organisms we barely notice here at home. Fungal enzymes are used to make biodegradable plastics, synthetic rubber, and even Lego bricks. The same molecular tools that fungi evolved to break down plant cell walls in forests are now being harnessed in industrial processes to manufacture materials we use every day. Fungal enzymes can synthesize polymers at lower temperatures and pressures than traditional chemical methods, reducing energy costs and environmental impact. Companies are experimenting with fungal processes to create everything from plastic bottles that degrade harmlessly after use to rubber compounds for tires, even the colorful interlocking bricks that generations of children have played with. It is a quiet revolution in manufacturing, replacing petroleum-based chemistry with biological processes refined over millions of years. Fungi can be grown into leather-like materials and sustainable building products. Mycelium, when grown under controlled conditions on agricultural waste, forms dense mats that can be harvested, dried, and processed into materials with properties similar to animal leather, supple, durable, capable of being dyed and stitched. Fashion companies are already producing bags, shoes, and garments from fungal leather, offering an alternative that requires no animal raising, no tanning chemicals, no greenhouse gas emissions from livestock. Beyond textiles, mycelium can be grown into forms and shapes for construction, insulation panels, acoustic tiles, even structural components that are lightweight, fire-resistant, and completely biodegradable at the end of their useful life. Mycelium-based packaging is being developed as an alternative to styrofoam. Agricultural waste, corn stalks, sawdust, hemp fiber, is inoculated with fungal spores and packed into molds. Within days, the mycelium grows through the substrate, binding it into a solid form that perfectly matches the mold shape. The result is packaging material that cushions fragile items during shipping, but unlike petroleum-based foam, can be composted in a backyard pile where it will decompose in weeks. It represents a fundamental shift in how we think about materials, not as permanent products, but as temporary assemblages that return to the earth when their purpose is fulfilled. Fungi are being used to convert agricultural waste into biofuels. Crop residues, corn stover, wheat straw, sugarcane bagasse, contain enormous amounts of energy locked in cellulose and lignin. But accessing that energy requires breaking down these tough polymers into simple sugars that can be fermented. Fungi, with their lignin-degrading enzymes, can accomplish what chemical processes struggle with, converting waste biomass into feedstock for ethanol production or other biofuels. It is a way of closing loops, of ensuring that nothing is wasted, that the stubble left after harvest becomes the fuel for next year's planting. DNA studies reveal thousands of unknown fungal species in every handful of soil. When scientists extract DNA directly from soil samples and sequence the genetic material present, they discover vast numbers of fungal sequences that don't match any known species in databases. These are called dark taxa, organisms detected by their genetic signatures but never seen, never cultured in laboratories, never described or named. They exist in a taxonomic shadow, known only as strings of nucleotides in data files. What they look like, how they live, what roles they play in their ecosystems, all of this remains mystery. It is humbling evidence of how much remains unknown. How many residents of the soil community operate entirely outside human awareness? These are called dark taxa, fungi that have never been seen or named. The term evokes their invisible nature, their existence in darkness, both literal and metaphorical. They may be rare, existing in tiny populations that escape sampling efforts, or they may be abundant, but simply unlike any fungus we have learned to recognize and study. Some may lack conspicuous fruiting bodies, spending their entire lives as microscopic filaments. Others might fruit only under specific conditions that rarely occur, or in seasons when no one is looking. The dark taxa remind us that biodiversity is not just what we can see and count, but includes vast realms of life that persist beyond the reach of our names and categories. Fossil evidence suggests fungi existed on Earth over 800 million years ago. Chemical signatures in ancient rocks, particular lipid molecules that only fungi produce, indicate their presence in neoproterozoic seas, hundreds of millions of years before plants colonized land. These early fungi lived in water or perhaps in shallow marine sediments, already employing the same fundamental strategy of external digestion that modern fungi use. They witnessed the Earth transform from a planet of mostly single-celled life to one where complex multicellular organisms began their long experiments with new forms, new strategies. When we look at a mushroom today, we are looking at the descendant of organisms that have persisted through nearly 1 billion years of planetary change. Fungi have survived multiple mass extinction events. Through the end Ordovician extinction, through the Great Permian dying when 90% of marine species vanished, through the asteroid impact that ended the age of dinosaurs, fungal lineages persisted. They may have even thrived in the immediate aftermath of catastrophes, decomposing the vast quantities of dead biomass, flourishing briefly in landscapes emptied of competitors and predators. Their resilient spores, their ability to colonize disturbed habitats, their broad metabolic capabilities, all of these traits made fungi survivors when other kingdoms faltered. They are living links to Earth's deep past, carriers of genetic information that predates most visible life. They have quietly witnessed the rise and fall of countless species. While continents drifted and mountains rose, while forests spread and ice ages came and went, while trilobites gave way to dinosaurs and dinosaurs gave way to mammals, fungi were there, decomposing, connecting, recycling nutrients through ecosystems that came into being and passed away. They watched species emerge and diversify and go extinct, and they remained, adapting but fundamentally unchanged in their basic strategy. It is a testament to the elegance of the fungal design, to a body plan so versatile and effective that it has required little modification across geological ages. Today, fungi continue to support nearly every ecosystem on the planet. In rainforests and grasslands, in tundra and deserts, in freshwater streams and the ocean depths, fungi perform essential functions that other organisms depend upon. They decompose organic matter. They form partnerships with plants. They cycle nutrients. They provide food for invertebrates and small mammals. Remove the fungi from any ecosystem, and it would collapse within years. Plants would struggle without their mycorrhizal partners. Dead matter would accumulate. Nutrient cycles would slow to a crawl. They are the hidden infrastructure that makes terrestrial life possible, the unseen workers maintaining the systems upon which all other species rely. Without fungi, there would be no forests, no soil, and no life as we know it. This is not hyperbole but simple ecological fact. Trees depend on mycorrhizal fungi for survival, particularly in nutrient-poor soils. Soil itself is largely a fungal creation, the product of millions of years of organic matter broken down and mixed with mineral particles. The rich humus that sustains plant growth, the dark earth that we cultivate and depend upon, all of it was made by fungi, is being made by fungi still, in the ongoing slow work of decomposition that never ceases. Life as we know it exists because fungi make it possible, because these quiet organisms perform work that no other kingdom can accomplish as effectively. And so our quiet walk through the fungal kingdom comes to a gentle close. We've wandered through hidden forests of mycelium, across ancient networks woven in silence, past glowing caps in the darkness and spores drifting on invisible winds. We've seen how life depends on what we cannot see, how patience, connection, and slow transformation hold the world together. It's humbling to remember that beneath your feet, wherever you stand, there is likely a vast and patient network, a living web older than memory, breaking down the past and building the future, one quiet thread at a time. You are walking on top of an invisible world that breathes, senses, and responds. It reminds us that life is not just what we see in daylight, but what works in the shadows, in the soil, in the slow hours when nothing seems to be happening at all. If you've enjoyed it, you can gently like, subscribe, or leave a thought in the comments. But for now, there is nothing more to do. Just allow your body to sink deeper into rest. Let the day fall away behind you like autumn leaves settling on the forest floor. Let the darkness be soft and full, like the rich earth where mycelium spreads in silence. There is no need to hold on. Just let go and let yourself be held. Sleep well and dream beneath the forest. Good night.